The human body requires a variety of foods in order to maintain itself and to provide energy for the vital activities. Before these foods can be absorbed into the living tissues of the body, they must first be checked by mechanical and chemical actions in the digestive system. Digestion begins in the mouth, where food is mechanically ground by the teeth and mixed with saliva. The saliva is made by 3 pairs of glands whose ducts lead into the mouth. Here is exposed a salivary gland of an animal with a glass tube inserted into the duct. When I stimulate this nerve by touching it, the glands begin to secrete saliva. They are under the control of the nervous system. A little vinegar placed in the mouth produces reflex stimulation of the secretory nerves and causes saliva to flow. Foods activate the salivary glands in this way. To study the chemical action of saliva on foods, we shall take 2 identical suspensions of starch. I will now add saliva, which we collected from the man, to the starch suspension in the glass on the left only. The saliva of man contains an enzyme called ptyalin, which converts the starch into soluble sugars. We see the starch suspension on the left gradually becoming clear, as the ptyalin breaks up the large complex starch molecule into smaller soluble molecules of dextrins and sugars. If we examine food that has been masticated thoroughly, we find that the coarse food particles stick together. This is due to a substance called mucin in the saliva. I pour saliva into this dropping funnel on the right. And water into this funnel on the left. The mucin gives saliva a thick and viscous consistency, as we can see by comparing its rate of flow with that of water. Therefore, the salivary mucin helps us to swallow by holding the food particles together and by acting as a lubricant. The esophagus has an inner lining like that of the mouth and an outer coat of muscle. Here, the esophagus of an anesthetized animal is exposed. The movements of the esophagus in swallowing coarse food are clearly shown. The muscular tissue of the esophagus, shown dark in this diagram, is under the control of motor nerves shown in white. These stimulate successive parts of the esophagus to contract, thus causing a peristaltic wave. The entire nerve and muscle action is much more complicated than shown in this animation. The stomach is primarily a reservoir for food, but within the stomach the gastric juice starts to change the proteins. The inner lining of the stomach contains glands that secrete this gastric juice. This consists of water, mucin, hydrochloric acid, and enzyme pepsin. By swallowing a rubber tube and applying a slight vacuum, we can draw out some gastric juice and study its action on foods. To study the chemical action of gastric juice on proteins, I'll take an egg and separate the white from the yolk. Then I will take this tube, draw the white of an egg up into it, and drop it into hot water to coagulate the egg white. I will break the tube to a suitable length, place the tube with coagulated egg white in a dish, and then pour the gastric juice over it. As we observe this tube at 2-hour intervals, we see that the opaque solid egg white becomes dissolved or digested at both ends by the pepsin and hydrochloric acid in the gastric juice. The very large protein molecule is broken down in steps. The first products are large and insoluble molecules, The end products are smaller and soluble molecules. Stomach functions are essentially controlled by chemical and nerve factors working automatically. The movements of the stomach are peristaltic in nature and in the early stages of digestion sweep across the lower half of the stomach towards the outlet of the intestines. When food has been reduced to a semi-liquid consistency by the gastric juice, it passes into the duodenum. Here it is mixed with bile and pancreatic juice. The pancreatic juice is secreted through ducts into the duodenum. It is a clear, alkaline and somewhat viscid fluid. The activation of the pancreas is an intricate process. In the course of gastric digestion, hydrochloric acid is produced by the glands of the stomach. This acid, imagined here as white particles, passes with the food into the duodenum. Here, the hydrochloric acid stimulates the lining cells to produce a substance called secretin, imagined black, which is absorbed into the bloodstream. Then it is distributed to all parts of the body, including the pancreas. The secretin activates the cells of the pancreas to produce the pancreatic juice. A similar mechanism acts on the liver and gallbladder. The liver secretes bile, which is important for the digestion and absorption of fats. I shall add bile to both of these samples of cream. And I shall add pancreatic juice to the one on the left only. During the next few hours, the cream at the left containing the pancreatic juice gradually clears. Pancreatic juice contains several enzymes, among them lipase, the only digestive enzyme which has the power to split up fats. The complex fat molecule is broken up by the enzyme lipase into separate and soluble molecules of glycerine and fatty acids. The small intestines are supplied with blood vessels and nerves. Beneath the outer coat lie longitudinal muscle fibers. Below these is a layer of circular muscle fibers. The inside lining reveals a mass of small projections called villi. During intestinal absorption, these villi, photographed here through a microscope, make rhythmic contractions. These contractions probably help to stir the contents of the intestine and assist the flow of lymph. These villi absorb the products resulting from digestion. In this animated diagram of a single villus, we portray the sugars and products of protein digestion entering the bloodstream. Now we see the end products of fat digestions entering the lymph stream. Imagine how a contraction of the villus would force the lymph onward. This absorption of food products is helped by local movements of the intestine wall. A certain area containing food may show rhythmic constrictions and divide and churn the food. There is also another type of intestinal action. This is the peristaltic movement, which consists of an area of relaxation followed by an area of constriction. This action moves the food onward in the intestine. After spending several hours traversing the small intestine, the indigestible material or roughage, together with traces of food and much water, enters the large intestines. Here the material is moved along and much of the water is absorbed. The large intestines also show local rhythmical motion and the peristaltic motion, in addition to the mass movement which occurs during defecation or colon elimination. So we have traced the processes of digestion through the alimentary canal and have followed the digested foods into the bloodstream. The failure of these processes to function normally may result in serious malnutrition, while adequate food and normal digestion are essential to health and efficiency.